Okay, so let's start with today's session. I think, guys, you can just turn your mic mute. Okay, and even you can uh, turn your video off. I think somebody have just S Madhavan. Can you please mute your mic? Okay, so let's start with today's session. So we are basically going to discuss about nervous system today. You all are must aware of, and you must have gone through the assignments and theories. So today I'm going to mainly discuss about few basic concepts that you should know already, and I will discuss that, and you can just ask me if you want anything to get it clear. Guys, you should just join 5 minutes before, then it will be more convenient for everybody, right? Okay, so let's start. Okay, so basically we are going to discuss about nerve impulse conduction today. Okay, so here you can see in the figure, you all are, I believe that you all are either bachelor's or above qualification students. So most of you are aware of this diagram that I am going to show you, or that currently you are seeing in your screen. Okay, so so nerve impulse conduction is basically the transmission of stimulus or signal that we are going to discuss, then how it is happening. Okay, and what is the electrical characters of different ions, or how it is regulated basically throughout the nervous system. Okay, so if you find, just I would like to make you aware of three things. Okay, so how many of you are aware of the resting membrane potential? At previous class we have discussed one point that just a minute. Okay, so previous class we have discussed that there is a basic contraction is already present in the muscle, right? So that is called your minimum amount of contraction that stores energy always for future purpose. Okay, similarly this nervous system or these neurons have a basic amount of stimulus at resting potential as well, that is around, you must are aware of, like minus 70 millivolt something, right? Okay, so now if the resting potential is minus 70 millivolt, then there are some sort of regulation inside the neurons that maintaining that particular resting temperature, or resting potential, right? So there are three sources, or I would like to say three potential channels, or you can say three potential sources that manages all these transitions of different ions and maintain that resting potential. Okay, and that threes are your sodium channel, potassium channel, and sodium potassium pump. Okay, now what is the difference between channel and pump? Any of you just aware of? Okay, just let me discuss that. See, channel is regulates singly at a time. Okay, one channel can just open up inside, one channel can just open up outside, but pump, okay, pump is a system that always act. First of all, it needs energy. Okay, and the second thing is that it work in both ways. Okay, it transfers something outside, in returns it transfers something inside. Okay, so pump are one that maintain the balance in both direction. Okay, if it is a pump, then it will releases something outside of the membrane. Suppose if it is in the membrane, if it is releasing something outside the membrane, simultaneously it will take something inside also the membrane, and it will maintain the balance, and it needs energy. Okay, however, channels are one-way traffic. Okay, they transfer in a single direction, or they work in a single direction. Okay, so suppose one channel is open, that tends to move your some molecules, for example sodium, inside, then it will only move sodium inside the plasma membrane. Okay, if potassium channel is open and it's supposed to, it opens outside, then it tends to move potassium from inside to outside only. But the sodium potassium pump, what it will do, it will act in such a way that it will take 3 molecules of sodium outside and 2 of potassium inside. Okay, so in that way it acts in bidirectional manner. Okay, and all these 3 works simultaneously to manages this resting potential stage, that is your minus 70 millivolt. Okay, now for example, let's imagine what should happen in the membrane system. Okay, see, there are so many proteins you will find inside the cytoplasm, right? And you all know proteins are generally negatively charged. Okay, so inside the cytosolic environment is generally negative. Okay, so you will find the outside membrane is basically positive due to the abundance of your sodium ion. Okay, now the point is that if sodium ion is so many sodium ion is present outside, and internal environment is comparatively negative, then there is one molecule, that is potassium. So as per the concept, you will find outside is positive in terms of sodium ions. Okay, inside is negative. Now in order to manage that negative amount, potassium ion is present inside the membrane. Okay, now that potassium ion has a tendency, or everything has a tendency to move from higher concentration to lower concentration. Okay, so we'll see, in order to balance those proteins or this negative cytosolic environment inside our membrane, potassium ion is present, and that potassium tends to move from inside the cytoplasmic membrane to outside because of the concentration gradient. Okay, and that happened by the leaky potassium channels. Okay, so you will find the potassium ions can travel easily from the inside membrane to the outside membrane. Okay, sodium cannot move that easily. Okay, so overall this sodium channel, potassium leaky channel, and sodium potassium pump manages a resting stage of minus 70 millivolt. Okay, now suppose this is your resting stages. Okay, so there is no norm impulse conduction right now. Now suppose there is a stimulus. Okay, and what happened if a stimulus is given to the membrane? Okay, now at the point the stimulus is given, you will find in figure B, at the point the stimulus is given, sodium channel open inside. Okay, so you will find sodium channel open, and sodium channel allows all the sodium molecule to move inside. We already discussed that sodium concentration outside the membrane makes it positive. Okay, however potassium concentration inside makes it negative, comparatively negative, because sodium is bivalent and potassium is univalent. Okay, so you will find that the sodium channel opens and sodium moves inside. The point or the moment it gets a stimulus, that defines or that progresses the negative ions outside. So negative or electronegativity, it increases the electronegativity outside the membrane. Okay, because of the only sodium channel. So in stage 1, from the resting potential to the action potential, the things that plays a major role is your sodium channel. Okay, so it is the sodium channel is the master molecule or master structure that define and that depicts the entry of sodium ion inside the plasma membrane, and that makes outside the membrane negative. Okay, and inside retrospectively positive. Okay, because if outside is negative, then the sodium ion comes inside and it will increase, or comparatively it will make positive. Okay, now the third point is that if it is depolarized, that stage is called your depolarized state. Okay, because the thing is at rest, that is called resting potential, and you will find some yes, somebody have raised your hand. Would you like to say anything? Okay, so it is from the resting stage the polarization get depolarized. Okay, because the sequence that the membrane inside is negative and outside is positive is altered because of the stimulus, and sodium ion get inside, and that changes results in the change into your starting of action potential. Okay, and that is when it is happened at some particular level. Okay, now 2 point is there. Okay, first point is that there is a stimulus. Okay, so there should be a threshold of the stimulus that makes the membrane to work, or that makes the membrane to act, and that energy is called your threshold potential. Now, for example, you will see, suppose just take any example. Suppose there is just a minute. Yeah. Okay. Suppose there is a boundary. Okay, and you are trying to jump that off. Okay, so it is your limitation, or it is your limitation that some people. Yeah, this is 5th week assignment only, or I'm not sure rather, but yeah, I think 5th week assignment. Actually, can you just tell me what you are studying in your 5th week assignment? What you have questions? Because these are previous year questions, so that's why. Can you just tell me? Anyone? Guys, can you just tell me what questions you have? Like, are you in your 5th week? Are you studying neuroscience only, right? Yeah, right. Yes. Okay. See, actually, we are not allowed to discuss this time question. Actually, I was even initially I was discussing this year question only, but later they told us that we are not allowed to discuss. Okay. Okay. So so it was supposed to that we are expecting that you guys are only having the questions from this areas. Okay. That is what I can say. Or even apart from your answers also, you can just listen to me. Just I will take your 1 hour and you will get some ideas. Okay, so this is what I was talking about that. Okay, so this is what I was talking about, that there is a limitation, and that defines whether how many of you are able to cross the boundary, and that is defined by a particular limitation or minimum criteria. Okay, so that is basically your threshold energy. Okay, so that threshold needed to be crossed in order to perform a action potential. Okay, so that is defined by two ways. That is the strength of the stimulus, that it should be efficient enough to create action potential, and the second thing is that the amount of potential that is created inside the nervous system or inside the neurons should have the ability to travel. Okay, should have the ability to move forward to give a successful stimulus. That also depend on the initial point, that is the amount of the stimulus that is given to that particular neurons. Okay, so now once the membrane is depolarized, now it tends to repolarized. Okay, it is, once the stimulus is there, now the stimulus will move forward. Okay, and once it will move forward, the position that initially get depolarized needed to revert back into its initial position, and that is what called your repolarization. Okay, now how it will get repolarized? Okay, so in order to repolarize, there are two things. The first point is that the activation of potassium channel. Okay, now what potassium channel do? It can only transfer the potassium ions outside the membrane. Now see, it is very critical or interesting if you will find that, see, if only potassium ion travel outside, then also the membrane potential can be managed. Okay, but for example, if you get another stimulus, okay, if you get another stimulus, then can it be possible to initiate another stimulus? No, because there is no sodium at all outside. Because see, during depolarization your sodium came inside. Okay, but during repolarization, suppose only potassium channels are activated, it will manage the ionic strength, or it will manage the electropositivity or electronegativity. But if only potassium ions get revert back to outside and manages the potential, then if another further stimulus activated, then at that particular time the signal cannot be processed, because for depolarization sodium channel is essential, right? And if there is not at all sodium present outside the membrane, then how can the sodium channel can channelize the sodium molecules in order to process further stimulus? Okay, so now it comes the role of sodium potassium pump. Okay, so the sodium potassium pump, what it does, it transfers 3 sodium outside in exchange of 2 calcium, 2 potassium inside. Okay, that manages the sodium ion concentration outside. Okay, so that if further stimulus started, then the sodium ions that are evicted out by the sodium potassium pump can again get inside by the help of sodium channel. So I will make you crystal clear that it is the sodium channel that results in the start of action potential. So in order to maintain, so once sodium channel get activated, sodium will get inside, and if sodium are inside, then it needed to be get revert back, and that is done by your sodium potassium pump. Okay, so that if further stimulus get activated at that particular time, that sodium ion can again initiate another stimulus in order to perform further nerve transmission. Clear? Now the next slide is also just like that only. So if you'll find here, see, there are several stimulus that as I discussed. So minus 70 is your resting potential at which this membrane is at rest. Okay, now there is a point called threshold stimulus. So it is the minimum amount of stimulus that initiate an action potential. So once that action potential reached, then only, unless and until it reached till the action potential, your nerve impulse conduction will not take place. Okay, so it is bound to have the minimum amount of threshold in order to start action potential. Okay, now that goes up to plus 40 to plus 30, okay, even plus 45. After that, your further repolarization occur. So yeah, repolarization occur by the help of efflux out of your potassium channels. Okay, and if potassium get outside, then the potential will again decrease down till minus 70. Okay, but due to the leaky channels, what happened, some more amount of potassium get evicted out, and that results into your refractory period. Okay, that reaches up to minus 80 to minus 90, that is your refractory period. Okay. Okay, so based on that part, we are going to discuss few questions. Okay, so the question number 1 is that the resting membrane potential of the neuron mostly is around, here we see, right, the resting membrane potential is around minus 70 millivolt. Okay, the second question is that when positively charged sodium ion flows inside the cell, the potential rises in and is called rises in which direction and is called what? Okay, I already explained you that when positively charged sodium ion flow inside the channels due to the action of sodium channel, okay, in which direction? That is in positive directions, and is called your depolarization. Okay, because sodium channel is the structure that initiate the flow of sodium inside the membrane, and that results in the depolarization. Okay, and depolarization leads to your action potential. Okay, now the third question is that the efflux of which ions assist in membrane to resting membrane potential. Okay, now the membrane is depolarized due to the movement of sodium channels. Okay, suppose the membrane is now depolarized. Now which ion will assist? Okay, as I told you, if membrane is already depolarized, that means that sodium ion is inside and potassium ion is outside. Okay, so now it is the potassium ion, okay, that will get effluxed out, okay, by the help of potassium channel. Okay, so as I have already told you that depolarization is started with the sodium channel, however the repolarization is managed by the potassium channel. Okay, so that is called your repolarization. Okay, now the next slide is that we have few more questions. Yeah, so for action potential to occur, the potential need to shoot below 100 millivolt or should above minus 30, or it's like that. So it is basically asking you that for action potential to occur. Okay, so in order to start a action potential, I told you that there is a minimum amount of stimulus to require, and that is called your threshold. Okay, so as we have already discussed, so it is started from minus 70, right? So from minus 70 millivolt, it needs to be, it is at least here, it is, you can see minus 55. Okay, so it is the minimum amount of stimulus that leads to the action potential. Okay, so this is basically based on that question that it is above minus 70. Okay, here if you will find your options, so it is like below minus 100 millivolt, above minus 30, remains between minus 60 to 70, that is your resting potential, or remain at resting membrane potential. So both rest three options except this particular one are just tends to tell you about the resting potential. Okay, but this is what, that the above minus 30. Okay, this seems to mean that it is starting from minus 70 and it will go still minus 30 or minus 50. That is the minimum amount of stimulus that leads to form your action potential. Clear? Okay, now the question number 5th is that the sodium potassium pump exchanges how many potassium in replacement of sodium? Okay, I told you that it exchanges 3 sodium ion outside and 2 potassium inside. Okay, channels are always in one-way traffic. They are only tends to work in one direction. However, pump are bidirectional and they act that they often do under active transport. Okay, that means they need energy, and energy currency is your ATP, right? Now this sodium potassium pump releases out 3 sodium in returns of 2 potassium inside. Okay, Now the question number 6 is that the sodium potassium pump, how many decks sodium and what it happens to the potassium. Okay, so I already told you that it will export the sodium ion, and because see, during depolarization sodium ions get inside the membrane, right? Now sodium is inside, so what pump can do? Now why you will use energy? Energy is always used when action is happened against the concentration gradient. Okay, see, sodium ion concentration is already outside is very much. Okay, suppose if you want to transfer some amount of sodium inside the membrane, then you don't need energy, right? But if you want to transfer sodium ion against the concentration gradient, then you need to have active transport, and that is done by your sodium potassium pump with the use of energy. And what it does, it exports the sodium ions and imports your potassium ions. Okay, now one more concept is your saltatory conduction. Okay, now what is saltatory conduction? Okay, so saltatory conduction is a mode of north impulse conduction where you will find there are nodes of Ranvier. Okay, if you will find the place where the nodes are there, these are your nodes of Ranvier. Okay, if my crosshair is visible to you. So generally what happens in the neuron, there are the blue color, if you will find, these are myelins. Okay, the myelins are basically lipid. Okay, and lipids are they are very active in this conduction of the signal. Okay, so in here what happens, the north impulse does not pass through the entire membrane. Okay, rather it will pass through only the portion of the membrane that are not covered by your myelin sheath. Clear? Now if that is the condition, that it is not going to occur in the place where myelin sheath is covered, so if the majority of areas will cover with the myelin sheath, and so the north impulse will only pass through the node of Ranvier. Okay, node of Ranvier means the position where there is no myelin sheath. Okay, so what it will do, it will directly jump the signal. Okay, if you can find, if you see, you can see the figure. Initially you will find there are negative outside and positive inside. Okay, so that the potential or active potential is started. Okay, now that will directly jump to the next node of Ranvier instead of the membrane that is covered by your myelin sheath. This process is called your saltatory conduction, or the process in which the axon potential jumps from one nodes to the another nodes. Okay, that is basically called your saltatory conduction, and that is, it is only seen in the myelinated actions. Okay, myelinated actions are basically covered by myelin sheath, and myelin sheath is basically one lipid that covered the nervous neurons, and it helps in the first transmission of your nervous signals. Okay, now this is what your question is. The jumping of north impulse from one node of Ranvier to the another is called your saltatory conduction. Okay, that is means jumping conduction, you know, that it is not going to pass through all the neurons, rather it will only pass through the portion of the neurons that are covered by your myelin sheath, that are not covered by your myelin sheath. Okay, now the next question is that which cells are used to myelination of nervous system. Okay, so the myelin sheath are formed by your Schwann cells. How it forms, you can see here. Okay, so Schwann cells are mainly present in the axon. Okay, if you find the axon here, you can see, or you the neuron, you can see the Schwann cell is present. You can see here with the nucleus is also present in the Schwann cell, and that form the cover, you know, sheath. Okay, and that is basically made up of myelin, and myelin is basically made up of sphingomyelin. Okay, so this is the myelin sheath. Okay, this is the myelin sheath that is formed by the Schwann cells. Okay, so this is basically covered form around the Schwann cells, and this is mainly made up of lipids and help in the saltatory nerves conduction. Okay, now the sphingomyelin in myelin sheath is here what it is. It is basically a lipid. Okay. As I told you, sphingolipids are composed of three things. Okay. One is your sphingosine backbone. Then what is sphingosine backbone? So sphingosine backbone, if you will find in the structure here, so in sphingosine backbone you will find there is one alcohol and one phosphate is present, okay, along with R group, okay, and the special and one long chain fatty acid you will find the C after the C, the long, this is your long in white you can see this is the long hydrophobic tail, okay, and one polar head, okay. Polar head is basically made up of your sphingosine, okay. Now if you'll find this structure in details, just I would like to show you this thing, that it is basically made up of your hydrocarbon chain, one fatty acid and one phosphate group, and the R group is your choline, okay. So in myelin, the R group in the sphingolipid is your choline molecules, okay, and this is what comprises or a lipid structure, that is your myelin, and this myelin help in your formation of myelin sheath around the neurons, and that helps in the process of saltatory nerves conduction. One more concept is that the minimum length of nerve signal that can elicit a biological response. So what is the minimum length of nerve signals? Okay, so minimum length of nerve signal, what you are getting, just let me clear this concept to you guys, okay. So there are two terms as shown here, okay. One is Riobase and one is Chronexy, okay. Now what these two terms basically means to you, okay. So Riobase kya hota hai? What is Riobase? So this is the minimum current, okay. This is minimum current, that means it is a stimulus. So it is the minimum amount of stimulus that is required to depolarize a nerve given at infinite duration of time, okay. So suppose I am providing a stimulus for infinite amount of time, then also it is the minimum current, okay, that is, it is the minimum current, that is, it is a stimulus, that is, it is a threshold, okay. So that is the minimum current that depolarizes the nerve, okay, if it is provided for infinite direction. Now what is Chronexy? It is the duration of current required to depolarize the nerve threshold when the current is 2 times of the Riobase. Now initially I told you that Riobase is the stimulus, that is the minimum stimulus, okay. Riobase is the minimum stimulus that can stimulate if it is provided for infinity time, okay. Now Chronexy is the double of the Riobase. It is the duration, it is the time interval that can be used, or that is the frame that is used to generate twice the Riobase amount of current, okay. So the duration of current required to depolarize a nerve to threshold when current is 2 times the Riobase. So it is the duration, okay, it is the duration when 2 times Riobase current means here, means is this, that if it is 1 time Riobase, it is a stimulus, okay, that is for infinite time, it is going to just activate, or it is just going to stimulate. Now if you are giving 2 time Riobase current, 2 time Riobase means 2 time amount of minimum current, okay. So if it is the twice of the Riobase, and this current, how many time it will take to how many time it will take to depolarize the membrane, okay. So that time interval is called your Chronexy. So Chronexy is the basically the time interval that is required to depolarize when the current is double the Riobase, and Riobase is the minimum amount of current that is required to depolarize a membrane when provided for infinite time, okay. Now your question is that what is the minimum strength of the nerve signal? So from this term only you will find it is the minimum strength of the nerve signal, means that is the stimulus, and the stimulus is your Riobase, okay. However, if it is the double the Riobase, that stimulus, how many time it will take to make a signal or make a stimulus, that time interval is called your Chronexy, okay. Okay, so guys, anything or any question that I should repeat? Yes. Yes. Yeah. See, suppose just let me give you a simple thing. Okay. Uh, if it is a signal, okay, if it is a signal, okay, then it is transferring, right? If it is a single neuron, okay, if imagine this, there is a neuron, okay, suppose this is a neuron, now this is dendrite, and you are asking that it is starting from the cell body. If there is no nervous stimulus that is associated with this, and to the dendrite it is only axon that is associated, right? That is you are calling, right? So if there is no axon, okay, because this is of the previous neuron, it is not of the same neuron, okay, so if there is no axons in the previous neuron, okay, and if there is no stimulus, then the stimulus is not going to carry forward, okay, so that's why it is told that the stimulus started from the axonilla, okay, but in the same cell it is started from the dendrite only, and that is also you can see the new junction, where by the neurotransmitters your stimulus or your signals is transferred from the axon hilла to the dendrite of the next neuron. Okay. Is it clear? Yeah, sure. Yes. Yeah. Uh, multiple cannot, and it's also not like that key at the simultaneously both excitatory and inhibitory works cannot be performed by particular neurons, okay, so there are two type of neuron, one as you told, excitatory and inhibitory neurons. Yes. Yeah. Yes. No, you can just go ahead. Just that's what I'm telling. Ki at the same time, both of the neurons cannot perform their functions. Okay. So it's not happen generally. Ki at simultaneously you need to perform both. Either you have to perform excitatory function or inhibitory function. Okay. And the path is also different. It's not like that the path is similar, because excitatory function there is everything is different. Like starting from the neurotransmitter, the transmission of signal, everything is different as compared to the inhibitory, because the neurotransmitters that plays a very important role in such kind of signaling or transmission. So it is the neurotransmitter that will define, and at a simultaneously both of the neurotransmitter cannot activate at a nervous junctions. Okay, axon and dendritic in a junction, at a simultaneously your excitatory and inhibitory signals cannot active. Okay, so it is generally not happen ki at simultaneously both excitatory and inhibitory functions any neuron can perform. It's always have to be like, see, the time limit is very less, but they can perform one at a time. but what actually your question is? Addition of nervous from one neurons to other, or you are talking about the addition of excitation? Yeah, single signal, right? Yeah, see, actually the problem, actually the thing is that suppose one stimulus is there, as you just see, like from positive to negative, it comes till the end. Okay, so just if you can see just any neurons, if you'll find this neuron, suppose a signal is just transferred till the neuron end. Okay, now here is your neurons axon and dendritic junction. Okay, so now this what it will do, it will releases few chemicals that is called your neurotransmitters. Okay, that neurotransmitter okay, that neurotransmitter is going to bind to the receptor in the next neural dendrite rather. Okay, so that will change the nervous potential, that chemicals will change the nervous potential. Okay, so these are the neurotransmitter, like as you told, excitatory or inhibitory. It is not like that it is going to fasten the reactions or slow the reaction. It is the neurotransmitters that are basically the chemicals, and that are signalized by certain receptor present in the dendrites, that are going to sense that chemicals, and that will stimulate started the further stimulus, like whether it is excitatory or inhibitory. If it is excitatory, then it will move in other direction. If it is inhibitory, then again it's it will gradually like move. But the point is that the signal, that the charge you are talking about, right, and the charge, this like positive and negative, that is what we are considered as a stimulus, is transferred from one neural junction to the other via the neurotransmitters. Okay. Yes. Yeah, but the point is that yes. Go ahead. Okay. Yeah, two action potential cannot occur at the same time, but they are neither, even they cannot get added. Okay, it's also a point. But the stimulus, as you are talking about, if both excitatory and inhibitory are added to the same neuron, okay, then actually even we have to find it out. I don't think that even it is possible, because for this you can see, like if it can be a different way, like suppose one is bind to a dendrite, okay, then the other neuron is bind to another dendrite. Okay, so in that way that can transfer the signal. Okay, you can see that the path will be different, because there are so many dendrites present in a cell, right? So if that is the case, then that might be the reason, but actually we have to confirm, and then only I can tell you. Okay, so we'll see, we will discuss in the next class regarding this. Okay, is this fifth week is question answer is remembered, sir? What does that mean? Okay guys, so see you in the next class, and just actually I'm facing that issue, but anyway we'll have a discussion next class. Okay, or how many of you think like we can just discuss few more questions, like I can give you few questions like in Google Forms and all, like test. Okay, we'll see regarding that. Okay, we'll definitely consider all those things. Anyway, from the next class we'll see to make it more interesting and make you guys to participate more. Okay then, we'll see you in the next class. Thank you for attending the class, and even you can go and find it in YouTube and all. Okay, so no issue, but be in the class so that you can ask and come with questions. Okay, you go read, you have so many questions. Okay, just put forward and let's discuss, because that is more interesting, and that will even let me learn, let you learn, and let everybody learn and realize some interesting question. Okay, apart from discussing only these questions. Okay? So come with the question, and thank you so much. Okay, I will see you in the next class. Thank you guys.